right. So now that we looked at Robert Griffith and we saw that DNA was a transformed molecule, now we can go ahead and look deeper into what DNA is, how is it made, etc. So DNA is a monomer. If you remember monomer, mono means single, poly means many. So it's many being made up of singles. The monomer of DNA, the single, the brick of the brick wall, are nucleotides. And nucleotides have three parts: a phosphate group, a sugar, and a base. Phosphate group, we're not going to worry too much about this. You'll see this in chemistry. We talked about it earlier with ATP, but we're going to leave this alone for now. Okay, but just know it's really polar and it's there and it never changes. The sugar is deoxyribose. There's only two sugars we're going to mess with. We're going to mess with deoxyribose and then ribose. So this is the D one, right? That's what gives the DNA its D, is the deoxyribose, is the sugar. So as long as you remember that the D sugar is in DNA, you're fine. And the nitrogen base here is a part that changes. It's a variable group, but it's a very limited variable group, and you'll see why. There's only four bases: A, G, T, and C. Now, that's thymine, cytosine, adenine, and guanine. The good thing is they each started with a different letter, and the letter is actually how we remember the actual base. We're not going to get into the structures of this. So this little structural formula, there are some classes that will do it, just not ours. So that being said, we're going to jump into this and we're going to say that ATGC, right? The purines, right? The U has double rings, like a double U, right? Double Y doesn't make any sense, but double U does. So the purines, the U's, the double U's have double rings. And the pyrimidines, which are the Y's, they have one ring. So that's a way to separate them. It's just two classifications of bases. Leave it up to biologists to classify things when you only have four things total. This came about from the structure of DNA came about from these two British folk, Watson and Crick. They created the three-dimensional model of DNA, but a lot of their work was based on Rosalind Franklin's work. Rosalind Franklin took this picture of DNA, which is looking down the barrel of DNA. Now, I've had this structure of DNA explained to me from this picture a number of times with a number of things and showing me in different ways. And I can recount some of it, but what I don't understand is how she looked at this and figured it out, right? It's one thing to, it's kind of like being able to see the maze once someone shows you the answer, but she was able to figure out the maze, before any of that. So Watson and Crick took her work and then took it further. Now you might think, well, why didn't she do it? Well, she died at 38, right? She died rather young, so she may not have been able to uh, uh, do this kind of work. Plus, when she was doing this work, female scientists weren't exactly, a dime a dozen. They were very rare, and they weren't always taken as seriously as they should have been. But Chargaff also came up with this idea that for every A, there's a T, and for every C, there's a G. And this makes more sense when you see it uh, paired up. See how this A and this T are paired up. So green and red, well, orange, I guess. Let's call it green and red. So here's green and red, here's green and red, here's green and red. Every time you see a red, it could be red on one side or the other, doesn't matter. It's always with a green because A is always with T. Okay, and G is always with C. Purple, yellow, purple, yellow, purple, yellow, purple, yellow, purple, yellow. It could be yellow, purple, right? That doesn't matter. But they're always together. So I like to think of it like G and C, like the store. That helps you remember that G and C go together. Also, when you draw the letter A, you say you draw three sticks for A. One, two, three, and T are two sticks. The sticks stick together. It's just a way to remember that T and A go together and G and C go together. If you think of another way, you're more than welcome to. So here's T and A, here's A and T, here's G and C, here's C and G. They bind together. These are the bases. Here's the sugar and the phosphate. And then when you look at this actual bonding like this, you see that it's a DNA strand over here and a DNA strand over here. And if I go back, you twist that ladder and you get a twisted helix. It's like a twisted double helix ladder. And that's the structure of DNA. And that's it for section 2 of chapter 8.